Linking monomers to polymers entails the process called condensation, while breaking down polymers into their constituent monomers proceeds via hydrolysis. Both these reactions are relevant to the linear polymers we'll be discussing in the following sections. DNA is a linear polymer made up of 4 monomers called nucleotides. Each monomer contains one of 4 nitrogen bases, cytosine, thymine, guanine, and adenine, designated C, T, G, or A respectively. The base groups are linked to a carbon 1 of a central 5-carbon sugar called deoxyribose. Deoxyribose has free hydroxyl groups at carbons 1, 3, and 5. The hydroxyl at carbon 3 remains free, while carbon 5 is bound to an acid phosphate group. Hence the name deoxyribonucleic acid, DNA. Here are 4 simplified representations of the nucleotides of DNA, yet still show their salient features. Each has its nitrogen base, the free hydroxyl at carbon 3, and an active phosphate group at carbon 5. Nucleotides can be linked together through condensation, generating a chemical bond connecting carbon 5 of one sugar to carbon 3 of the next via the phosphate group, which bridges between them. Consequently, the linear polymer has a backbone made of alternating sugars and phosphates. The DNA has 2 distinct ends, the 5' end, referring to the phosphate at carbon 5 of the deoxyribose of the first nucleotide. The last monomer has the remaining free hydroxyl of carbon 3 and is designated the 3' end. So all in all, DNA seems rather simple, having only 4 distinct monomers linked via alternating sugar and phosphate groups. As it turns out, however, the composition of DNA proves to be more complicated, as was discovered by Chargaff. Chargaff, a biochemist, measured the concentrations of the nucleotides of DNAs taken from different organisms. He carefully determined the precise relative amounts of G, A, T, and C for each of the DNA samples he examined. Here is a table of his original results. At first, the numbers seem a bit confusing, and a pattern is not immediately obvious. However, Chargaff realized that the amount of A is always closely equal to that of T, and that the concentration of G seems to be the same as that of C. Moreover, if you compare the A plus T content to that of G plus C, for any organism, it turns out that each organism has its distinct and very reproducible nucleotide composition. Thus, for example, people have 1.5 times more A plus T than G plus C, while the bacterium E. coli is measurably richer in G plus C. But this is not a general rule for all bacteria, as the DNA from the bacterium Clostridium is remarkably poor in G plus C. Ultimately, Chargaff formulated a set of 4 rules, commonly known as Chargaff's rules. 1. The DNA composition for a given organism is defined, constant, and reproducible. 2. This DNA composition for different animal species varies. 3. The DNA compositions for a given species does not change over time, with age, or from organ to organ. And finally, the most important rule, the concentration of G equals that of C, and A equals T. These pair ratios, G to C and A to T, always equal 1. Clearly, DNA has a unique composition, but how it functions as the stuff genes are made of is a whole different story.